Good morning, everyone. Thank you for joining BrainMap today. Before we start, I want to remind you that this BrainMap Seminar Series is co-sponsored by the P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. It is my pleasure to introduce our today's speaker, Dr. Elizabeth Hutchinson. She's an assistant professor of biomedical engineer at the University of Arizona with research interests that span both imaging science and neuroscience. Her lab uses advanced imaging approaches for development and understanding of novel imaging markers of brain changes in neurological disorders, particularly in traumatic brain injury and Alzheimer's disease. Her educational background includes a bachelor's degree in physics from Penn State University and a PhD in neuroscience from the University of Wisconsin, as well as a predoctoral and postdoctoral training at the National Institutes for Health in the area of advanced preclinical neuroimaging. Her current research takes a multi-scale and multimodal perspective to advance translational neuroimaging tools for the understanding, diagnosis, and treatment of brain disorders. Today, she will talk about microstructural MRI validation and translational studies to improve detection of neuropathology following brain injury. The stage is all yours.
Thank you. Thank you for that introduction. Let me go ahead and share my PowerPoint here. All right, let me know if you cannot see that, but I'll assume that you can. Done this.
We see it.
Great. So thanks for having me. It's really nice to speak to this audience. I think there are a lot of things that we sort of have in common using microstructural MRI to study and advance diagnostic techniques for brain disorders. I thought I would focus on two aspects of this sort of larger field that are of interest to this group and to me certainly, which are validation and translation. So with that in mind, I'll begin. Our common goal, I think, among sort of all of us and many scientists in the area of advanced imaging is to improve diagnosis and prognosis through advanced techniques for imaging. Particularly, this is through MRI, because MRI itself is very well suited to doing this. I like to show at the beginning of classes that I teach that the Nobel Prizes that are awarded in our field, the basics of MRI, were initially in physics in the earliest part, and then moved to chemistry, with NMR. But the Nobel Prize in magnetic resonance imaging, that is forming an image using these principles, was awarded in the area of medicine. And I think it's really important to highlight that the ability to visualize soft tissue is fairly unique to MRI. And it makes what we do a real opportunity and somewhat a unique opportunity to look at all the organs of the body, but in particular the brain. And we know that this is used clinically every day to afford many different types of contrasts that can let clinicians know the prognosis of a particular patient. This is, for example, cancer. And I think, within this, there's a real push to advance beyond just tissue contrast and be able to use some advanced sensitization of the MRI paradigm and also mathematics for quantitative MRI. And hopefully this will more and more make its way to the clinic, but it's going to take a real effort. So within that sort of big umbrella and that common goal, microstructural MRI is sort of one of the most active areas and one of the most promising ways of advancing towards the goal of improved clinical diagnosis across many types of brain disorders. And so I just sort of start by showing where that fits in to the larger scheme. Most people are used to the sort of human brain images and at the resolutions on the order of a millimeter. The work I'll talk about today is all preclinical, and it's largely fixed tissue. And so, a mouse brain's on the order of 10 millimeters. And if you wait a long time and use 3D pulse sequences and things like that, you can get down to a few tens of microns in resolution. But that's not really what I mean by microstructural MRI, as many of you know. It's more that we can look inside the voxel. So this is simply human connectome data, and probably the last time I'll show a human brain in vivo anyway. And the idea here is that when we're able to use quantitative techniques to look inside the voxel, we extend our length scale of sensitivity into the microscale range. And this could be potentially very important for understanding and developing new markers for brain disorders. This is particularly the case for traumatic brain injury, which is what I'll be focusing on. And I'll just go through these sort of cartoons that relate what's really truly a big bloody organ that is the brain into what's happening at the cellular scale when the brain is injured. We know that what the neuron's made of, the dendrites, the axons, and so on. And that we also have astrocytes, about half the cells there. Now, when we undergo a brain injury, many things happen that physically change the cellular environment very prominently. This can be axonal being, hypertrophy of neurites, loss of neurons, loss of myelin, and so on. And then the glia can change, they can become reactive, or you can have infiltrating microglia that change greatly in their, physical sort of morphology. And so this is why microstructural MRI can be so promising for looking at this. The definition extends beyond just diffusion, which I'll be focusing mostly on today, to other techniques as well, and hopefully I can touch on some of those. There was this really nice special issue in Neuroimage that many of you are probably familiar with, that I think gives you a nice sort of overview of the microscale features that we're sensitive to, cells, dendrites, things like this, and the techniques that we can use for them. That said, there are a lot of challenges to microstructural MRI, and all of those sort of clinically relevant images I showed on the first slide, really not many of them are these techniques. And our goal should be to get these techniques to become more used clinically. So part of the challenges for using microstructural MRI are that they can often be difficult to implement. The acquisition paradigms are specialized and the processing pipelines are challenging. And then you always hear this lament that diffusion especially is very sensitive, but not specific. And so we really need to better bridge the scale of our maps with the scale of the cells. And then, the methods can also take a long time and it's hard to get them into a clinical scanner. But I think if there was a will, there'd be a way. So if we can really demonstrate that some of these techniques have great clinical promise, then I think it would be manageable. So today I'll talk about two areas that address these. Validation studies are sort of targeted at this sort of middle point of sensitive but not specific. And then translation studies are really targeted at finding what is the most promising to translate into clinical use. Before we talk about those, though, I think we need to introduce the microstructural MRI methods, the ones that we primarily use in our lab, and also talk a little bit about the effects of image quality and acquisition parameters, assumptions, and artifacts. I spent a lot of time as a staff scientist in Carlo Crepelli's lab. A lot of that work was really geared towards understanding also how imaging features can affect the metrics that we report, because this is equally important to understanding how the biological features can affect those, because we want to be free of artifacts and bias. So I'll begin by talking about the microstructural MRI methods and then move into validation and translation later. So from that same sort of special issue in Neuroimage, they did a word cloud of everything that was included in all of the articles that sort of represent what we think of microstructural MRI as today. And diffusion is the largest word because, as I think many of you probably think, diffusion is almost synonymous with microstructural MRI. Not always, but there's a good reason that people think this way, and that so many of the microstructural MRI studies are diffusion based. And that is that there's this sort of very elegant relationship between water diffusion and brain tissue. And so it's the self-diffusion, this long history of Perrin, who got the Nobel Prize for it, and Einstein, who came up with some of the math to describe water self-diffusion or other particle self-diffusion. And we can really leverage this, and that's what we do, to be able to probe what's happening on the microscale to our cells in our brain. And Cheshkul and Tanner were able to sensitize the MRI scanner to be sensitive to this, right? Using the B value as a diffusion weighting metric, and then measuring the signal at different B values. You're all probably very familiar with this. From there, we can then represent the apparent diffusion coefficient, which is dependent upon the tissue environment instead of the physical properties, right? So this is really useful in lesions. So you can see in this bottom case is following middle cerebral artery occlusion in a mouse, and I'll always put the species of brain in case you're a human person up at the top right. And what you see is that in a region where the tissue is lesioned, we of course have less steep decay because diffusivity is restricted and ADC is low. And so we're able to very prominently map that. And it's sensitive to something that a simple T2 weighted image is not. You cannot see the pathology here, you can see it here. And this was, sort of the jumping off point for the experiments by Mosley et al., which showed that even in the absence of T2 changes, we see diffusivity changes. This is in a cat after a stroke, right? And so I bring this up because it is probably the most used clinical technique that applies microstructural MRI. Now, of course, this has extended to the diffusion tensor, in which you can sensitize the scanner to diffusion along different directions. And I have a bonnet macaque brain shown from a collaboration with Carol Barnes in Arizona. And you can really nicely see in the white matter of this brain sort of the changes due to the restriction in some directions or hindrance of the water in some directions, but not others. And of course, we get the diffusion tensor, which is in the scanner reference frame, and we can get our eigenvalues and take it out of the scanner reference frame and come up with a number of metrics that tell us about the three-dimensional sort of magnitude and shape of water diffusion, in a very sort of basic way through the diffusion ellipsoid. And we use these fractional anisotropy and trace or mean diffusivity maps to characterize that. Now, those models are 20 plus years old, the diffusion tensor model is. And since that time, it's been noted that there are a number of limitations with that model that if we can just overcome, we can, have a better characterization of tissue and get at that sensitive, excuse me, but not specific question, right? So the first limitation is that it cannot capture complex geometry. So this is a ferret optic chiasm. And you can see FA, there's an FA map, FA is low in the middle. And that's not because the anisotropy of the fibers is lower, it's because of crossing fibers. So this is the crossing fiber problem. And if we move to higher order models, we can more adequately capture those crossing fibers, right? But the diffusion ellipsoid alone cannot. Okay, also we're limited to where the signal behavior is sort of follows an exponential decay with diffusion weighting. But above a pretty low B value nowadays, you'll have deviation from this due to non-Gaussian signal behavior, which is due to restricted compartments or other aspects of the tissue that we might be interested in. So there's a lot of potential if we can look beyond a Gaussian decay model. And finally, as with many of these physical techniques, it lacks biological specificity. So, even with higher order techniques, we get something that looks like one of these ODFs, but we don't really get something that looks like neurons in vasculature. So something that I've been interested in for a while now, and this is work from when I was in Carla's lab, is what are the sort of benefits and costs of using these non-Gaussian diffusion models? And so I'll go quickly through one of the papers we have on this, looking in sort of a TBI model. This is in ferrets, just to compare what these different metrics get. You might think that we can get a bunch of different stains, and that could be advantageous if they are telling us something different about the biophysical environment. Okay, so the sort of next step from a DTI model is the kurtosis model, where we just sort of add a term. Unfortunately, this is a quadratic term. So the shape of the diffusion kurtosis curve is very important, and only certain data will support that. We get maps of mean kurtosis, kurtosis FA. An extension of that is mean apparent propagator MRI, which extends this to sort of arbitrarily high non-Gaussian shapes. This was pioneered by Evren Ozarslan and Peter Basser's lab, where we can fit the signal in Q space and use the same coefficients to represent the propagator. There are a couple nice properties of this model. The the 0th order is actually the diffusion tensor itself, and then anything above that is non-Gaussian. From that come a number of metric maps, including non-Gaussianity, which compares the full propagator to the 0 order term, the return to origin probability, which is a nice propagator metric sort of in general, because it's just the propagator at 0 displacement and tells us the probability that a water molecule hasn't moved. And this can be useful if we're thinking about restricted compartments. If we had a whole bunch of fully restricted compartments, this would have exactly the inverse of the pore volume of those compartments. So it's a nice metric to look at and think about. And finally, the nice thing about propagator models is you can compare propagators. And so if you compare the anisotropic and the isotropic version of this, you can get a metric called propagator anisotropy. And this is that same ferret optic chiasm. And what you can see is that in the region of crossing, you would have you wouldn't have the same signal dropout due to the crossing fibers. So it's thought that propagator anisotropy represents more the microscale anisotropy of tissue. And this is the one that I'll focus on today because I don't have time to go into all of them. But when I bring up higher order models, this is propagator anisotropy, so I'll talk about. I also just want to briefly mention that biophysical models are a really important microstructural technique because they may give some relationship between our diffusion signal and the actual biological tissue. NODI is probably one of the most popular and common ones. It uses a tiered approach and models the intracellular and extracellular space as cylinders and sticks to give different volume fractions of cellular and intracellular spaces, as well as an orientation dispersion index, which is shown here. So these are the main tools that we use right now to look at different changes in brain injury and in neurodegeneration. There, of course, have been many more since this paper came out, and we're always sort of trying to add to the toolkit. One can really sort of in a more general sense idealize this as having a way of collecting sort of data that supports a wide toolkit of stains to look at different outcomes after an injury or in a neurologic disorder. And this is really useful at the preclinical level, particularly. But we have to make sure that they're worth it. So if they don't tell us anything new, or if they're so hard to use that they're never going to be translated, then enthusiasm is diminished. And so one of the things that we've done that I just want to talk about quickly is we've looked at the dependence of these different models and techniques that I mentioned to image quality features and to diffusion encoding. And I'll just, for time's sake, focus on diffusion encoding. So we looked at whether sort of a low B value encoding scheme versus a moderate versus a high in Black affected different metrics in the same way or differently. So for example, you're probably familiar with diffusivity shifting to lower values due to noise floor. Also, if you sample out to sort of this non-Gaussian range of B values, you will have a systematic bias in diffusivity towards lower values because you're now sort of calling restricted water hindered water, and it's no longer Gaussian. So this is sort of an example of systematic bias. And we looked at this across the models that I mentioned and found sort of a wide range of sort of different behaviors. And this is in mouse brains of these whole brain histograms, with the systemic bias, FA was fairly unchanged, and so on. And so you can sort of reorganize these into categories where you have relatively stable features of the different metrics. You have systematic bias, which can make conceptual sense. And then you have some that have considerable to severe vulnerability. And generally speaking, the more complex your model is, and potentially the more specific the information it gives, the more vulnerable it is to these types of biases. And I want to just point out propagator anisotropy and non-Gaussianity both show sort of a bimodal distribution when they're highly sampled. And the reason for this is because they really don't become all that useful until you have a B-value sampling that will support them. And so this is a major consideration, especially if you're strapped for scan time. Okay. But all of this may be worth it if we can use these methods to show us something that a standard DTI cannot. Right, so this is a case of that where we have fractional anisotropy in the ferret brain near an injury site, and we see that FA is completely gone. But when we look at histology, what we see is that there are remaining axons that stain positive for myelin, despite some damage and gliosis. And when we look at propagator anisotropy, just shown here, what we see is that it tells us something different than than fractional anisotropy. And this is really what we're looking for when we're comparing these higher order metrics. Is sort of the maintenance of sort of some type of metric information in a region that DTI might fail. So I'll just touch on briefly the other techniques. Since I've moved to Arizona, I've extended into also relaxometry-based techniques. The two that we use most are multi-exponential T2 decay for myelin water fraction mapping. So this is based on a fairly old work where the sort of biochemical composition of different cellular compartments can be seen in their T2 signatures. So you sort of have 3 peaks here. And this shortest one corresponds with myelin, and you can use this for myelin water fraction mapping. Of course, in studies of Wallerian degeneration, this part goes away. And so it's very useful potentially for looking at demyelination, which is a more specific outcome. Additionally, we use bound pool fraction mapping, which is acquired using magnetization transfer, which you're all familiar with, using pre-pulses that sort of saturate the bulk water and then partially saturate the bound water. And we can use some math to determine the bound pool fraction and map it. So, and then we can choose our inversion recovery times in a way that we are sensitive to the sort of exchange rates that we would like to map in our bound pool fraction maps. And so this is again the bonnet macaque brain, and what you can see is the type of data that we have been acquiring here, where we have echo times, as low as 6 all the way up to 200, and I think there are like 30 some of these, and we use those to generate T2 maps, and then also these myelin water fraction maps. And we use the REMI toolbox, which is from Kevin Harkins and Mark Does at Vanderbilt. And then we also similarly use the REMI toolbox to map bound pool fraction using this inversion recovery time. And this is just the data that we have and use. One of the things that Laurel in my lab has been doing is to look across these images, I think about a dozen of these bonnet macaque brains for part of a larger study. But one of the things she's looked at is the relationship between fractional anisotropy and myelin water fraction and fractional anisotropy and bound pool fraction in different regions. And as you might expect, they correlate in the white matter, where FA and myelin water fraction are strongly correlated, but bound pool fraction in the white matter is not strongly correlated with fractional anisotropy. And then in the hippocampus, neither metric is correlated with fractional anisotropy, suggesting that there could be complementary information in these techniques. We've also used this to look initially at some brain injury outcomes. This is actually a fairly severe brain injury where we expect Wallerian degeneration. We presented this at ISMRM in 2020. And what we did was we ran these types of maps, and you can see that there's an increase in some regions in T2 and in T1, but a reduction in bound pool fraction and myelin water fraction in other regions. Now, these brains were taken 1 week after injury. And so later we would expect for, Wallerian degeneration to result in demyelination, but this is a way we haven't seen this yet. And then you can also compare this to fractional anisotropy, which doesn't just give you myelination information, but also axonal information as well. Okay, so that was sort of a whirlwind tour of some of the methods that we use and some of the things that we're interested just in the imaging science part. I'll now talk about validation and then finish up with translation. So validation to me is a really messy thing to try to do. It's very necessary to bridge what we're able to measure with what is happening at the cellular scale. But there are a lot of challenges, and I just want to go through this example of that, where we have in fixed mouse brains, we have found sort of these very, very prominent reduction in diffusivity 1 day after injury that shows here on the voxelwise map, and then this sort of increase in fractional anisotropy at 4 and 12 weeks after the injury in the cortex, following a mild but still penetrating cortical impact using a CCI device. And we see the same thing in vivo when we follow these animals, this dip, in this increase in FA. What we really wanted to do is, and this was work from when I was in Carlo's lab, is to correspond these with histology. And it seemed like a really easy thing to do initially, but turned into sort of this more monumental challenge than we had appreciated initially, right? So we have, these are the 4 brains at 24 hours. They show this reduction. It looked like there were 3 zones, but there's a lot going on here. There's gliosis happening, sort of microgliosis in the core, no astrocytosis yet at this time point. And then we have beating of neurites in the surrounding area, and then healthy neurites beyond that. And then it changes completely, where we have some of the same imaging outcomes, right? But at 4 weeks after injury, what we really have is this increase in FA and this sort of core of decreased diffusivity that corresponds to a number of co-localized histologic changes, right? And so, we've actually like submitted this work 4 times and it's gotten rejected 4 times and I'm done with it. But it's I think a real problem with validation, because I don't know that we've come up yet with the best way to do this. And so I have the question of how can we improve our ability to run these radiologic pathologic correspondence studies. And one, which I think a lot of people always suggest, is to move to quantitative methods. And we'll talk about that. And the other that, I would suggest is equally important is to take a phenomenological perspective of sort of things that we already know happen. And I'll just give 2 examples of those. Actually 3 examples. The first examples are like a really clear-cut case. So sometimes, as in this, you are using double diffusion encoding to try to map an axon diameter distribution. And you can look at the histology and you can get, a ground truth axon diameter distribution. And this is something that a graduate student in my lab, Courtney, did on the histological side. And we collaborated. This is a study by Dan Benjamini and Peter Basser's lab, where, we sort of just provided some of the histology and the tissue. And what we found was that, you can get these types of axon diameter distributions by doing some fairly simple things in MATLAB. And this is very relevant for what we're actually measuring using a very sophisticated imaging approach. So to me, that's a clear-cut case of where quantitative histology can really contribute to validation. Another way I think that histology is being used quantitatively is for data-driven studies. So along with Margalit Haber and Ramon Diaz-Aristia, Carlo and I looked at FA decreases following the closed head inertial injury model in mice and found that regions of damage corresponded to decreases in fractional anisotropy. And the way that we validated this really was by optical density measurements in specific regions of interest across a wide variety of stains. And we were able to sort of show that diffusion shows about the same thing. Instead of running a correlation analysis, though, you can take this same type of quantitative data and feed it into a machine learning classifier algorithm. In this case, this is a random forest analysis, and ask that to classify TBI versus not TBI based on your inputs, and come up with either classification error, and histology performed only a little bit better than diffusion in this study, or which metrics are the most important for prediction and so on. And that data is shown here. So this is sort of the other way I think quantitative imaging of histology can be useful. That said, I think that it's not the only way forward. And I think a lot of times we don't always look at the sort of body of literature that exists already in neuropathology studies. And I want to give just one example of that sort of perspective on doing validation. And so the basic paradigm is that there are sort of basic research studies that show some cellular phenomenon that accompanies some type of, usually animal model pathology, but sometimes validated in humans. This then sort of summates and contributes to a body of knowledge, right, where we understand the phenomena of the neuropathology that evolves, say, after an injury or in a particular neurologic disorder paradigm. And then we can use that, sort of build on that foundation of knowledge to identify either in a sort of hypothesis-driven way, go out and find them, or to qualify what we see when we just sort of look for imaging markers, to identify potentially relevant imaging markers. And so I think that this approach is well characterized by this story of mossy fiber sprouting. That comes about from Tom Satula's lab. I was a postdoc there in neurology a decade ago. But he did these basic research studies, actually, like when I was in middle school, I think, and showed that there's this phenomenon of sprouting of the mossy fibers in the dentate gyrus, so that the dentate gyrus cells end up projecting onto their own sort of input fields, onto their own dendritic layers. And this establishes a recurrent excitatory loop, which leads to seizures, and it's a focal seizure generator. And that this can be measured using what's called TIM staining, which stains specifically for these mossy fibers. And there's a scoring method associated with the presence of these axons from the mossy fibers in the dendritic layer. And that's what's going to set up this sort of recurrent excitatory loop. And that this TIM score can be associated with after discharges. And so this is sort of the basic research that set off this sort of description of mossy fiber sprouting. Now, since that time, it sort of contributed, along with many other studies in the field, to a more complete understanding that there's a loss of interneurons in the hilus, and that this can give rise to this sort of sprouting features, and these have been characterized morphologically, as well as compared with electrical recordings, electron microscopy, and so on. So then, to bring this sort of body of knowledge toward identifying, in this case, diffusion markers of mossy fiber sprouting, what we can do is sort of notice what those fibers might mean. I'm sorry, fibers, what the MRI outcomes might mean, and then go back and we need to validate them, using what we expect in a hypothesis-driven way to perform sort of informed histologic studies. And so this is something we saw, again, about a decade ago, which is that unlike control hippocampus, there's sort of this orientation change in the hilus of animals following canic acid administration. So this is an epilepsy model, pure epilepsy model, but also following TBI. And we thought, well, maybe what's happening is that, in the control case, all of the dendrites are sort of oriented, in this way, so that they're green, sort of in the dorsal ventral plane, and then in the hilus they are not. But with recurrent, mossy fiber sprouting, that morphology changes, and you get this sort of dorsal ventral orientation, which you can see using diffusion. And so, wouldn't that be nice if we have a diffusion marker of mossy fiber sprouting? But to do this validation study, and it just got accepted this week to JNEN, we had to take sort of a really intentional approach where we did the imaging at different time points. So we did at the immediate time point to know, is there just sort of a physical pulling on the tissue that could cause that change? Do we see it when we don't expect mossy fiber sprouting, because it takes about a few weeks to a month to emerge? Or do we see it later? And what we did see was that the timing was consistent with the temporal profile for mossy fiber sprouting, right? And then we did a series of histologic analyses. Some of them were semi-quantitative, but for the most part, they were informed by, the neuropathologic review and blinded students and so on, where we see apoptosis at 24 and 48 hours. We don't see much gliosis, which would be a secondary or alternative explanation for that outcome. We don't see that at 4 weeks. But we do see a very subtle increase in TIM score that correlates to sort of this orientation abnormality that we found. And so that was a lot more work than the random forest analysis. But I think it sort of just highlights the difference in that type of study, where you can look at a phenomenon that's occurring that accompanies a neurologic disorder, and intentionally look at the MRI markers that might be useful for measuring. So I think there's probably a place for both of these. And I think, I bring it up to this group because I think it's a good open question, right? And I also, through all of this, think we should maybe reconsider histology as the gold standard. The reasons for that are it's very specific. If you think about what you're actually doing, you're staining like a protein. And so you might miss something, or you might overinterpret what you're seeing. Also, it's very difficult to make direct comparisons, registration, processing. And it's also completely ex vivo. So I think what's been really nice is to see all these intermediate methods. Like Carla Miller's group has done a really nice job with polarized light imaging to look at the orientation validation and different tractography type methods and so on. And there are a lot of sort of emerging different techniques out there that could be applied to this problem. And so I think that that's a really promising direction to go, and something that we're interested in, certainly. And then finally, I would say, can we reasonably compare all of these ex vivo things? Like, even most of what I do are these ex vivo studies, but can we really compare that with what's happening in vivo, right? And so the sort of last thing I want to say about validation is that I think it's useful if we can come up with ways to do in vivo validation. And that's not unreasonable. People have done that for fMRI, certainly, and using 2-photon imaging to look at the hemodynamic response, and so on. And there's already even 2-photon, I think, of, the acute microglial response, I think that's Tori and McGavern, in brain injury. So, I think there's the potential there to use that for microstructural MRI as well, although we'll probably have to move beyond just 2-photon. Related to that is some of the more recent work that we've been doing at the University of Arizona, and this is with Kaveh Laksari and Paulo Payers, and our labs sort of have all gotten together as soon as we were able to do experiments again, and looked at different MRI and also photoacoustic and ultrasound outcomes immediately following controlled cortical impact in mice. And so we did sort of this MRI battery and compared controlled cortical impact here. So you can sort of see the hemorrhage and a slight reduction in diffusivity and larger reduction in blood flow to a pretty big and better characterized more targeted outcome of subacute arachnoid hemorrhage, which you sort of insert a filament and pop a blood vessel and see a pretty big outcome. The part of this that I think is really relevant for in vivo validation is that you can, in the same experimental paradigm, the same animal, use a non-invasive technique that can give you complementary information. And so in our case, we use photoacoustic imaging where you shine a laser light in, and then it creates some very local thermal changes that result in an ultrasound wave out, and you can detect that with a transducer. The advantage of this is then you can get whole brain images of anything that absorbs light and emits sound, reasonably. And that includes this top row, which is oxygenation maps. And I know these are hard to look at when you're used to looking at MRI. It's been a really big adjustment for me as well. But it does give us oxygenation information, which we don't get with MRI, right? And then you can also get laser Doppler, or I'm sorry, Doppler, ultrasound Doppler to measure blood velocity, right? And so then we can start to compare these things across the different modalities. And in this study, a lot of it's focused towards the blood flow, but it is interesting to compare diffusivity, which we don't see as widespread, with things like oxygenation. So in the TBI model, we have a modest reduction in oxygenation. We don't really see a widespread reduction in diffusivity, but then in the more severe subarachnoid hemorrhage model, we see this sharper reduction in oxygenation, and we do see this change here. And so that sort of ties back into the potential for a phenomenological description, especially for the hemodynamic outcomes and what's driving diffusion changes. All of this is to say, I think that there are a lot of promising cross-modal or multimodal modalities that can really advance us towards in vivo validation, which I think is an important direction to take. Okay, I think I have a little bit of time left, and I will spend that time talking about our translational studies. I'll start by talking about the work we've done primarily at the NIH and with Carlo's lab and Sharon Giuliano at the Uniformed Services University. And the basic idea there is that if we can have a better way to look at and understand traumatic brain injury, then we can really impact a number of people who are affected by it. Many of you are already familiar with this paradigm. And so the basic idea is to take the human injury and migrate it to a preclinical model. And so the assumption there is that what occurs in the brain of that preclinical model is the same thing that occurs in the brain of the human. And that is what we are trying to model, of course. The rodent is the most commonly used species for these types of studies, but all of the studies that I'll talk about here are in the ferret. We think there are a lot of strong advantages to moving these models into the ferret. Particularly, you can notice that its brain is folded. That's sort of the first thing. It also has relatively high white matter volume compared to the rodent, not quite as high as human. But high enough that you have a complexity of white matter. You have U fibers, you have crossing fibers, much more than the rodent, combined with the gyrencephalic cortex, makes us a much more anatomically similar brain to the human. But the animal itself is small enough to fit into our preclinical scanner, so we can scan it in vivo. And then, it's not a primate, so you don't have to move all the way up to a non-human primate model. Ferrets are mustelid. So we run the ferret diffusion experiments in 3 different models. The first is controlled cortical impact. And again, that's where you sort of make a cranial window and use an impactor to make a very, in this case, mild injury. There's no cavitation. And so what we found was an increase in fractional anisotropy that was most prominent, during the chronic period, and changes in trace that also emerged with injury. Okay, I think the most important thing about this to say is this is like completely different than what we saw in pretty much the same exact experiments in rodents and mice, where in the rodents we saw this increase in fractional anisotropy in the cortical tissue, but not much change in white matter. In the ferret, what we see is no real increase in fractional anisotropy in the cortex to speak of, but this decrease in the white matter. And so I think that the ferret might be more human similar in that way. We know that FA reductions are an important potential outcome in human TBI as well. We've also looked at BLAST, and this is also published this year. And this is largely with Sharon Giuliano, who did a lot of very beautiful histologic work to show different aspects of the vascular injury itself. And we were able to see with high resolution, just T2 star weighted imaging in the fixed tissue specimens, some vascular abnormalities that were consistent with what she was finding. That said, the control animals aren't entirely free of those. We did look, but we haven't published it. I don't think it's really mature enough to publish, but we did look, and I'll show it here because I think it's, sort of mature enough to show here. Some changes in diffusion outcomes, not in the whole brain, but just in the white matter, some reduction in diffusivity. The red lines are each one ferret in these different regions, and the red lines are blast, and the black lines are control. And then when we move to the higher order images, it's possible that there's sort of this increase in return to origin probability, which again is related to restriction, especially in the white matter, and a decrease in the propagator anisotropy. And so I think a lot more work's needed to be done on that, but it's a promising indication that there might be some outcome that we can measure with the higher order models that we can't really measure with diffusion. Okay, and the final model that I'll show is an inertial model. And so a closed head injury model of engineered rotation acceleration. This was developed by Cheryl Wellington and her lab and Peter Krypton, and they sort of have adapted it to ferret for us. And basic idea is a piston hits a helmet-like interface. It doesn't protect the head, it just makes you not damage the skull. And then the head is accelerated forward, and we actually have a strap that sort of pulls it back. And we just have submitted this work and looking at the diffusion outcomes. Now, what we did in this, instead of going sort of the ex vivo microstructural route, is we really tried to mimic what happens in the clinic and what has been identified by the community to be important outcomes related to traumatic brain injury using MRI. And so we tried to mimic the human common data elements, which include tier 1, just contrast protocols, tier 2, which include diffusion, as well as T2, quantitative T2, and then a vascular arterial spin labeling outcome. Now, the main findings of this, I think we had probably hoped that we could get a white matter mild TBI model, but what we really got was more like a whiplash model, I would say. And, I think that that's still interesting and worth reporting. What we saw primarily was that the milder or less biomechanically loaded cases didn't show anything terribly conspicuous. But there were 2 very severe cases that showed prominent hemorrhages that were localized primarily to the brainstem. And we really did not find many or any hemorrhages in the sort of brain and white matter here. And then we did look at diffusion, and there's sort of some modest initial decreases in fractional anisotropy and modest initial increases in diffusivity that resolve quicker than the FA changes. I think the more interesting thing, though, that we saw was on a conventional scan where we injected gadolinium contrast, and we looked at T2 flare and also at T1 weighted images, and saw that 3 hours, but not at baseline or later, we saw this sort of brightness of contrast enhancement in the meninges, either around the brainstem or in the midline. And this is consistent with what's beginning to be noticed in humans, as well as sort of this acute meningeal damage that shows up with gadolinium contrast and occurs in sort of a large number of patients. So the ferrets might be useful for looking at that as well. And I think the ferret has a lot of human similar brain features. And the more we can use human similar brain paradigms like BLAST or like Chimera, I think the more translational our studies can be, the more clinically relevant. And at the same time, I think using MRI outcomes that are already being used in the clinic is a really important, it's not always the most fun because we want to use the most advanced microstructural methods, but it's also, I think, a really important way to conduct these types of translational experiments. So, but what about those advanced microstructural MRI methods? I still think it would be really useful to decide which of these are the most promising to translate to the clinic. And so to do that, to say, are these outcomes that we're seeing in the ferret relevant for the human? And if so, should we then maybe consider pushing that towards clinical implementation? I think post-mortem human tissue offers a lot of promise for being able to do that. And I've been involved in these types of experiments for quite a long time, including some very nice brainstem studies that I started with Carlo, just in the normal brainstem, and also in cranial nerve disorders. We're able to use the advantages of ex vivo MRI and combining it with diffusion to map out very small structures like the cranial nerves, as well as their nuclei. And so when towards the end of the time when I was at the NIH, a postdoc named Mahika Gangoli joined, I forget actually whose lab, but she brought with her some postmortem data that had this really well-supported sort of study that she had published on just DTI, and I looked at it and thought, wow, we should really be doing, some of the higher order things on this data set to see if maybe we're seeing some of the same things that we see in the preclinical studies. And so I was really sort of eager to collaborate with her and also with Peter Basser's lab to look at potential differences between propagator anisotropy and FA and other things too. But like I said, I'll just emphasize propagator anisotropy. And it was pleasantly surprising to find that there was the same pattern of reduced fractional anisotropy, but not reduced propagator anisotropy in her samples as well, and that to some extent it correlated with GFAP. Furthermore, propagator anisotropy was more significantly correlated with her quantitative GFAP findings. And again, so this does two things. One, it says like maybe this is useful for telling us what's important in our ferret models. And then also maybe this is important for developing towards the clinic. So in my time at Arizona, I've sort of migrated from traumatic brain injury for postmortem studies to Alzheimer's studies, and started collaborating with Tom Beach, who runs the Banner-Sun Health Brain and Body Donation Program, as well as Gidi Serrano, to look at human brain tissue specimens with Alzheimer's disease. And I know I need to wrap up in the next 2 minutes, and I will. And so long story short, what Courtney Comrie in my lab has been able to do is to image these using a sort of series of different microstructural MRI techniques, and show, here's again that propagator anisotropy example that's different, where you see this bimodal distribution in the same voxels where you see a unimodal distribution of fractional anisotropy, and these are just hippocampal voxels. And then when we look in the Alzheimer's disease, what you can see is there's sort of this preferential change in one of those peaks. It's easier to see if I overlap them, where there's sort of this reduction in this overall peak, but a different behavior overall than FA. And we're seeing this across different modalities. It's been interesting to put together, and she just finished scanning about a dozen samples. So that's sort of where we are with that type of perspective on translational studies. Okay, I just want to wrap up. I think the microstructural MRI methods are, they're really an important way forward for neurologic disorders and clinical diagnosis and things like that. But we really need to focus a lot on a better validation studies. Moving that to in vivo would be fantastic. Just having a better way to look at comparing histology and imaging would also be fantastic. And then translating these, understanding what should be translated, what's relevant, how our animal models relate to it, I think is also of the most importance. And I know we're about at time, so I'm going to wrap up by thanking so many people. A lot of the sort of review work that I showed was with Carlo Pierpalli and all of the people in his lab, and that's been remarkably educational and important time spent there. And then also in the new lab here, I have wonderful grad students in Laurel and Courtney, and everyone else in the lab as well, and also collaborators and funding. And so I'm going to go ahead and stop here. And if anybody has questions, I will be happy to take them.
Thank you, Elizabeth, for this great talk. Yeah, so if anybody has any questions, just please go ahead and type them in the Q&A, or also just raise your hand. So while people think about their questions, I have a few. So my first, I know that you brought this up already, but that would have been one of my questions about how can we actually, like, if you can comment more, I guess, on the use of the ex vivo diffusion MRI for either validating or just investigating TBI mechanisms, because a lot of things change from the in vivo to the ex vivo brain. And so from the reduction of the extracellular compartment to, I'm not even sure, like edema, how that like translates to ex vivo. And so this is, yeah, always one of the things that stops me from actually using those values from the ex vivo. So yeah, if you can just, from your experience, comment more on this.
Yeah, I think it's really a challenge to use the ex vivo data. I think it's useful to compare it to histology, both are ex vivo, and some of the metrics can be useful. I think people have looked at the correspondence between FA, for example, or the directions. Directions, I think, probably don't change very much, between in vivo and ex vivo. Diffusivity, on the other hand, I don't think so. I mean, we see increased diffusivity in vivo, and then we'll look at the same brain and we see a decrease in that region. I didn't have much time to spend on that. In regions of edema with gliosis as well, you won't get any extracellular information in your ex vivo images, but that's really what's driving it. So I think there are things you can do. There's nothing you can do on the ex vivo imaging side of things. You could improve the quality of your in vivo imaging or the measurement strategies. There are plenty of techniques that take out free water or, measure different tissue pools and things like that. But I think the biggest thing is to actually just keep it in mind when you're doing a study, and not sort of say you see this thing ex vivo and that it should immediately be translated for in vivo use. But yeah,
Yes.
I agree.
You also probably use them for what we think that they can inform on and not use them for other things, I suppose. I don't know.
Well, or just do both. I mean, it's always nice if you can do both in vivo and ex vivo, and advance that field as well, because I think, there's more need for that too.
Yeah, so I guess your answer like leads me to another question, which is, so you said there's techniques to remove free water, sure, but most often those techniques cannot be applied to the data that we already have that come from the clinic.
Yeah.
And so I think that's another big problem, especially probably for the acute TBI cases where there's no advanced diffusion modeling you can do on that. So I don't know, I think out of curiosity, my question is whether in your career you've ever seen a situation where you use an advanced diffusion model and you validate it and you thought it was informing some way on the pathology, and that diffusion model and acquisition was actually used then and like transferred to the clinical. So that would be the first. And the second would be, and if you could actually do it, what would you pick?
Yeah, I mean, the answer to the first, I think often, I don't know how often it is that radiologists will even look at an ADC. I think they often will look at a diffusion weighted image, and there's nothing wrong with that. That gives that person the same information, right? So I think it's a real challenge to make the case that an advanced technique, and we spend so much time on these advanced techniques, to make the case that these should be used clinically. So I mean, to answer your question, I have not really seen that case. I have worked with clinicians on the postmortem tissue, and I think there's sort of a lot of interest there. I've worked with, surgeons trying a different approach in a brainstem or something like that, where they think diffusion could be useful. And then they could envision maybe using that same type of diffusion strategy in vivo. In the example I'm thinking of was from Wisconsin, and it's a brainstem surgery, which has, a very poor outcome, right? And so you could potentially use tractography in vivo for surgical planning and even the execution of a surgery, right? If you can learn from the ex vivo tissue, the right surgical approach and things like that. So I think, tractography's had a little bit more success, I think, being migrated clinically. You do see that for surgical planning and things like this. But as far as microstructural metrics that have a diagnostic value, I can't really think of one. I think there was someone at an ISMRM mentioned maybe prostate cancer was a good example in mean kurtosis, but I don't recall anything beyond that. So I would love to see something like, can we use directional changes in the hippocampus if that happens in humans? I would expect that to be true in vivo and ex vivo. And could we use that to predict after someone gets a traumatic brain injury, will they or won't they go on to develop, epilepsy? Because that's common afterwards. And so you could maybe predict that. So something like that, but it's a real challenge. And I think it's one that's important to like always have in our minds.
Yeah, that makes sense. All right. Well, thank you. If there are no other questions, I think we can thank you again for your great talk, and we can thank everyone for joining BrainMap today. And hopefully we'll see you soon, maybe in person at some point.
Thank you everyone for coming.
Yeah.